If you are a clinician scientist and would like to quantify the syndromic similarity between a cohort of patients, the research platform within GMDB will help you. In this video, you will learn how to use the research platform. At first, you need access to the research platform. To do this, you must first be authorized for this feature within GMDB. To do so, please send a request for your research project to info@gestaltmatcher.org. We will then activate your account for this function. If you don't have access to the GMDB yet, please watch our videos, sign up and access. In order to be able to analyze your cases with the GestaltMatcher AI, you first need to upload them to the GMDB. You can learn how to do this in our video Upload and Digital Consent. Alternatively, you can also work with the cases that are already available in GMDB. After that, you can start to create a project under My Research with a new research button. You can add a name for your project and also some notes. To select cases for your research, you have to return to the gallery view. Here you can search for your gene or syndrome of interest in the database and add all the images from this search to your research. It is also possible to add single images to the research. If you only uploaded your cases privately, they will not be visible in the gallery. You can find these patients under My Patients. Here we can also search for a specific gene or disease. The patients can be added to a research project here individually. To do this, a picture must be selected within the record of a case and added to the project. Under My Research, you can then edit the selection again and remove images that you would like to exclude from the analysis. You can also add a label to all the images. Don't forget to update your research after your changes. When you have added all cases to the research, you can start the analysis with the Run Analysis button. Depending on the number of cases analyzed, the process may take a moment. The results are then displayed in a pairwise distance and a pairwise rank matrix. This provides information about how close the cases are in the clinical phase phenotype space, CFPS, where distance is a measure of syndromic similarity. For more technical details about the CFPS, see the link below to our paper in Nature Genetics. To illustrate how clusters look like in the pairwise distance matrix, let's add some more cases of a genetic disorder with a different facial gestalt. The newly added cases with Chang-Jansen syndrome, which is caused by de novo mutations in PHIP, exhibit full eyebrows and/or synovial and often upturned noses as characteristic facial features. In contrast, individuals with B-allelic truncating variants in TMEM94 are characterized by a broad nasal root and tip and anteverted nostrils. The shape of the face is also more triangular. These differences in the facial gestalt result in larger distances in the CFPS between these two diagnoses, and separate clusters form if we sort the matrix by similarity. The pairwise distances of all cases in the high-dimensional CFPS can also be visualized on a 2D canvas by T-distributed stochastic neighbor embedding, TISNE. In the end, you can download the results. If you want to find out what else the GMDB can be used for, please watch the first video in our GMDB video series. If you want to stay informed about the GMDB features and new papers about the GestaltMatcher AI in the future, don't forget to subscribe to our channel. In the next video, you will learn how to interpret the results from your research in more detail.